Okay, class, so here's the lecture on DNA replication. So first thing I want to do is remind you of something. We did this a couple weeks ago, the cell cycle. Right? We saw the cell cycle represented with a pie chart. And in this pie chart, we had three things that were part of what's called interphase. There was G1, S, and G2. And then the rest of this, the small piece of the wedge, was mitosis and cytokinesis. And what this reminder is for is that in the S part of interphase, that's S is for synthesis, that's where we copy DNA. Right, so it's just kind of a reminder, like we have talked a little bit about copying DNA, which is what we're getting ready to do, right? DNA replication is just, we're going to copy the DNA. So this gives you the reminder of when would we be doing that and why. Right? Remember, this is cell cycle, so we do this whole cell cycle. We have one parent cell dividing to two daughter cells. So if we have one set of chromosomes and we're going to divide the cell, each daughter cell needs a full complement of chromosomes. So we have to copy all the chromosomes and then split them and then give them to the daughter cells. So this is a really important process, a really important part of what's going on in the cell cycle so that we can make sure we copy the DNA correctly. Okay, so then remember what we're going to do is take double-stranded DNA. So let's start with the picture like this. Here's our DNA double helix. All right, we're going to take that and we're going to separate the two strands. All right, so we've separated the two strands. And then we're going to build a new strand to go along with each old strand. So there's a double helix, and then here's a double helix. Right, so notice in my picture, like we're copying DNA, right? So like this is a logical picture. We have one helix, we copied the DNA, we have two helices, right? Now we've got two of what we started with, right? And notice that each double helix over here, sometimes these are called daughter, right? It's kind of common thing we use, daughter helices. Notice that each of these has one parent strand plus one new strand. Okay, so I'm going to show you as we go through this, I'm going to show you why that doesn't matter. Like we really are copying it, and it doesn't matter that there's one old and one new in each helix over here. All right, so that's big picture what we're doing with this. All right, so let's start with some DNA double helix here. So it doesn't matter what these bases or sequences are. So let's just do this. All right, so there's a piece of DNA. We know the complementary base pair. Right, so there's our helix. Right, so we can just say that this is our parent double helix. Right, that's what we're starting with in this picture. That's this thing here. Well, it's a piece of that thing there. Right, I can't show you something that's that long. Right, parent double helix. We know our complementary base pairs. So just like I showed you back here in this picture, first thing we did was separate the strands. So we're going to separate these strands. So it's easy enough to show. Separate the strands. Right, so we've separated the strands, and I think I missed something there. All right, there we go. Separated the strands. My G's look horrible. All right, we separated the strands. Now what we did up here was build a new complementary strand. Right, that's what this process here was building new complementary strands. Right, and we're going to use the original parent strand as a template because we know what our base pairs have to be. So we use those as a template. So if we start here to build a new complementary strand, we see an A, we put in a T. T, we put in an A, right? So complementary base pairs, building a new strand. Right, there's a new strand. And then we do the same thing here on the bottom. Use the black parent strand as a template and build a new complementary strand. All right, so then each of these now is a daughter helix. Right, so each of those are the two daughter helices that I showed you up here. Right, then each of those helices had a black strand and then either purple or green. So each of these helices has a black strand, which is parent strand, and then purple or green. All right, and to show you this, when we do this copying, we kind of want to make sure that, we really do make a real copy with no mistakes. So you can check that, right? You can compare this green strand to this black strand, and you can compare this purple strand to this black strand. And they should have the same exact sequences. So in here, using color, it kind of makes it look like visually that, hey, this is different. This isn't a real copy because it's different. But all that matters is the sequence, right? If you look at the order of the A's and T's and G's and C's, that's what matters. And the green and the top black or the purple and a bottom black, those are exactly the same sequence, and that there's no difference in those things. All right, so that's how what we're going to do when we're copying DNA, when we're doing DNA replication. Right, it's the idea we start with one helix and we end up with two. All right, so we use our complementary base pairs that we created, right? C and A, put a T, C, put a G, etc. And that gives us our two daughter helices. All right, the last thing with this is just, make sure that you can practice with this as much as you want. Like you can randomly make all the little DNA sequences, the little DNA double helix sequences like we did to start here. You can make all those you want and practice with this. What do I have to do to copy my DNA? Because you're going to see some questions that are going to be things like, if this is our parent double helix, then here's whatever, like what kind of question, some kind of question. Hey, you may see some questions, actually you will see some questions that are going to say things like, if this is our sequence, what's the complementary sequence down here? What goes in that other strand? So it's worth just taking some time and practicing with our base pairs and how we're copying and things like that. Right, so that's what we're doing with DNA replication. There's a lot of enzymes, and the book will talk about enzymes and all these steps, and I don't think we need to do that as long as we have a basic idea of what's going on with replication.